Hi, my name is Elsa Zhang, and I will be presenting on the effect of urbanization on green space soil. So what I'll be studying is urban soil, which are soils that have been extensively influenced by human actions. There are three types of this. The first is surface soils of surface layer greater than 50 centimeters. And why this is important is that if the surface layer is too large, that means like plants will not be really able to grow in that. The second is different composition, use, and management of conventional soils or agricultural soils. And third is soils that result from various construction activities. So all of these will result in soil that is not very great for plant growth and therefore affect green spaces negatively. And I will be doing this through soil percolation experiments and soil profiling experiments. Soil percolation basically just tells me how quickly water moves through a soil, which is important for evaluating the ability of soil to absorb water. Whereas soil profiling will tell me about nutrient management and as well as insight into soil fertility. And down here, you can see like the percentages of where sand, clay, and silt should be in the soil. And silt is actually the most important because most of the nutrients will come from the silt. And I conducted these experiments at the Eastwoods Neighborhood Park. So my question is, how does urbanization affect soil and green spaces? And my hypothesis is that it will decrease the nutritional value of the soil, indicated by long soil drainage times, meaning that the soil is too dry, and less than 40% silt in soil. For soil percolation, the materials used are a water stopwatch, shovel, and ruler. And how this works is that basically I will dig a hole. I dig the hole by about 5 inches deep by 4 inches wide with a shovel. And I poured a bottle of water, which is about 16 ounces, into the hole. And I timed how long it took to do that. So soil profiling is the second experiment and describes soil sample, dish soap, and jar of lid. So I took some soil from the sample and I put water and dish soap in it, and then I mixed it all together. And at different times, I marked like how much sand was in there, how much silt, and how much clay, which will give me the percentage of like the composition of the soil and tell me exactly how nutritious it is. For my results, for percolation, my drainage time was 1 minute and 20 seconds. After some calculations, that means the drainage rate was about 222 inches per hour. Profiling sand was about 25%, which is within the normal range. Silt is 30, which is a little bit low, and clay was about 45%. And at the right, you can see I included a picture of my bottle, and I marked down like about where the sand, silt, and clay are. So possible issues is that the depth of the hole for soil percolation is not that exact, meaning that although I tried my best to make a hole about 5 inches deep by 4 inches wide, it is not exactly like cylindrical shape, meaning that like the volume may have not been exactly like um, what I expected it to be, which could have messed with calculations. And for the soil profiling, the shape of the bottle was not consistent. As you saw, I used a water bottle, and there were a lot of crevices in it, which could mean that there's like varieties within the volume, and again, affected percentages and the distribution of the soil. So my conclusion is that since my drainage rate was over 1 inch per hour, which told me, according to a resource I found, would that mean the soil was well drained and suitable for all species, including sensitive species. So in this case, my hypothesis was rejected, meaning my soil was not actually too dry to support plant life adequately, as I'd expected. And a possibility of this is that, one problem that may have affected this part of it is that there's a difference in soil throughout the park. Like I picked one particular spot that I thought looked good, but in other parts of the park, it may have been really dry, and that would have given me like a different conclusion. Whereas, as opposed to this part, and I believe it was a little raining the day before, which could have affected that as well. And for soil percolation, the sand was normal again. There was not enough silt and there was too much clay, which means that my hypothesis was accepted or partially accepted. Because as I expected, there was not enough silt, meaning there was about, I think, 35% silt, which is not enough or to the accepted 40%. And there was too much clay, which can make it difficult for plants' roots to grow. So this indicates that it is possible that the nutrients are inadequate in the soil and which could affect green spaces. So some implications of this is this tells us that we need to protect green spaces because as you saw, like part of my hypothesis was indeed accepted. So possibilities for why this could happen would have been like maybe there was construction happening nearby, which affected that, or there were other factors due to urbanization that would have negatively impacted the soil and the green spaces. So we need to protect them because they're a vital resource for urban spaces. Erosion. So when there's too much sand or the composition of the soil is not the best it could be, like it is in ours, the possibility of erosion comes up, which could again negatively affect green spaces and the surrounding areas. But the drainage was good. So for perhaps for this area, it's not as big of an issue. And there were a lot of plants without their roots showing. So that was also a good indicator. And the third is climate change. So the biggest contributor to urban soil and um, is climate change, because with increasing temperatures and just overall like uh, extreme weather patterns, this has a really harsh effect on green spaces and is the number one contributor to urban soil, according to my research. So we just need to bring more attention to that, and this is something that affects everyone, including people in urban spaces. And thank you so much for listening to my presentation. Here are some of the resources I used that like relate to this presentation in particular.